So, good afternoon, everyone. So, for the next 5 minutes, I'll be talking about an engineering biology approach for PFAS management that encompasses all the three aspects to sense PFAS, to bioremediate PFAS, and the biosafety aspect, which is very important when we are talking about engineering. So, PFAS management is a uh, concerted effort where different stakeholders uh, take, can play different roles. When, as molecular biologists, we are exploring how engineering biology can, what engineering biology can offer in PFAS management. So when we are talking about engineering biology, apart from biosensing and bioremediation, the third factor, biosafety, becomes very important because we want to restrict the movement of lateral gene transfer selection markers to maintain the organismic homeostasis. So there has been many engineering biology attempts for PFAS management. For biosensing, some interesting reports are there where both protein-based as well as cell-based sensor have been developed. For biodegradation, although actual engineering biology approach has not been explored in that detail, but there has been significant groundwork we have heard in the morning session where genes and organisms have been identified, and those can be used in engineering biology approaches. Now, there is not much to talk about biosafety because engineering biology still is in early phase of development for PFAS management. So with this background, we propose a modular genetic engineering approach where we can integrate all the three aspects, bioremediation, biosensing, and biosafety, to create designer strains, to create designer microbe for better applicability of engineering biology in this field. So the design is composed of three modules. So the first one is sensor module that has a PFAS sensing promoter. Sensing promoter. It senses the presence of PFAS and relays the signal to the effector module. The second module is the effector module, which contains a reporter gene that provides an opportunity to biosensing, and the gene that can break down PFAS into non-toxic compound. And this effector module is activated by the recombinase from the sensor module. Now we have a third module, which is the containment module, that is important for the biosafety aspect. This module remains silent until the PFAS is present in the system. Now let's go to the modular crosstalk. I have put some animation to understand it in a better function. Under normal condition, there is no PFAS in the system. So the sensor module is in standby mode, and the effector module produces a repressor constitutively, and this repressor goes and binds to the module 3, which is the containment module, and it keeps the kill switch. The containment module has a lethal gene, which is a kill switch. So it keeps the kill switch silent under normal condition. Now, when PFAS comes, the sensor module activates. It produces a recombinase and the same repressor. So this recombinase goes to the effector module and clips the promoter. So the promoter which was driving the expression of the repressor, now the same promoter is driving the expression of a reporter and the gene involved in PFAS removal. Now the repressor is shut down, but still the containment module will remain shut because the repressor is coming from the sensor module. Now the promoter will produce the reporter gene as the gene and the enzymes that is responsible for PFAS removal, which will provide an window of opportunity for biosensing, and the bioremediation of PFAS will continue till PFAS is present in the system. Now, when the PFAS is gone, then the recombinase and the repressor is no longer available. Now, since the repressor is no longer available, the level of repressor in the cell reduces and the kill switch is expressed, which will ultimately lead to cell death. So that will ensure that after the PFAS is removed, the recombinant cells will be eliminated from the system. Now, as far as feasibility is concerned, most of the genetic elements are already available, and but the two main factor is a PFAS sensing promoter for module 1 and a PFAS sensing and a PFAS decomposing gene. So this article says our PRMA promoter, which have been shown to be inducible by P4, and it showed very weak level of expression, but we can make use of this promoter, since in this model, signal is getting amplified through this recombination based switching. So if we can explore more promoters, we can talk with the Princeton colleagues to explore more PFAS sensitive promoters and genes that can decompose PFAS. So what this design offer? First of all, it offers modular sensitivity where you can customize as per the requirement. As for example, if you don't need containment, then we don't need the module 3 to be integrated. Then this design offers a smart cell that can automatically sense, activate, and deactivate with a minimal intervention, and it offers signal amplification to overcome the limitation of native pollutant inducible promoters, and of course biosafety. It can remove the recombinant strains when not required. It operates through a biological NAND gate circuit, and of course, it can serve as a general template for other pollutants where you need to replace only the promoter in the sensor module and the enzyme that can break the pollutant. And these are some key challenges, like we have to have a PFAS inducible promoter that I already talked about. And leaky expression of reporter can be a spoiler. And it's a complex genetic engineering. Golden Gate and CRISPR can take care of that. And the burning question is multiple PFAS compounds. So it is highly unlikely that a single promoter can respond to all PFAS molecules. But even if we find a promoter that can detect maybe 2 or 3 or 4 most important PFAS molecules, so that could be a baby step towards a green solution. Thank you. So that's all from my side.